Be hot weekend. I don't know. I just called my brother in Florida and asked him how we do this kind of heat. Um, but he said stay inside and drink. I guess you'd have to know my brother. In any case, what we're going to talk about right now is chapter 5, which is cell signaling. This is the last in a series of three PowerPoints that are talking about cells. Where we were before is we talked about all the cell parts, how they work together, what their function is, what kind of processes go on inside the cell, for example, protein synthesis. And then we went to a chapter that talked about diffusion and the movement of stuff in and out of cells. This chapter is focused on cell signaling, and that is how cells receive information and respond to information, whether it be chemical or electrical. So that's what we're looking at here. When a cell responds to a chemical messenger, such as a hormone, for example, so if the endocrine system sends out a hormone to get a target cell to be able to do something that it needs it to do, that chemical messenger has to sit on a protein that is known as a receptor in the cell. Most chemical messengers are water soluble and they can bind to the receptors on the plasma membrane. A smaller number of messengers, like steroids, so we know that it is an example of a lipid. It's lipid soluble and it can bind to intracellular receptors in the cytosol or in the nucleus. So sometimes you have substances that are, and remember that steroids are found in the plasma membrane. Sometimes you have to have responses that actually begin inside the cell to stimulate the nucleus to do something, for example, to start creating proteins during protein synthesis, and those are the type of messengers that we're talking about here. The binding of a messenger to a receptor changes the shape of the receptor, which is then termed activated. So the sequence of events that respond to that ligand receptor connection is called signal transduction. And in our cell signaling lab, that picture that I gave you associated with the cell signaling lab, where sweet taste on a taste bud causes one signal transduction process, which is different than the bitter taste signal transduction process. So when we say signal transduction, we're talking about, in that case, a chemical receptor called a tastant, is what it was called in the lab, that attaches to the a chemical messenger, sorry, that attaches to the sweet receptor on the taste bud. And once that conformation changes, that shape changes for that particular receptor, then a series of events occurs to cause an electrical signal to be sent to your brain to interpret that taste. A typical transmembrane receptor is going to be made of that protein, that particularly folded protein that we talked about. And remember, in class, I went around and I showed that really the shape of this structure is the outside of all of this. If you were to trace all of this, that is the actual shape of that membrane. And when something comes and sits down in this particular site, it changes slightly the shape of this, causing the internal shape to start a series of steps that will send an electrical signal to the brain. So this is just like a close-up picture of what the inside of that protein looks like. Not all cells express the same protein receptors, okay? This selective expression leads to specificity in the systems. The response of individual cells with the same receptor also varies based on the cell type. So if this is an endocrine cell that is secreting a chemical messenger in the form of a hormone, different cells are going to express their protein on the picture that we saw previously that has the shape for that particular chemical messenger. If this tries to sit down in this type of a receptor, it's not going to bind, it's not going to change the shape of this, and it's not going to start the signal transduction. Same thing here. If this one tries to sit down into either one of these areas, signaling areas in the receptor, it's not going to fit, it's not going to change the shape of this, so it's not going to be able to transduce any kind of a signal into an electrical signal that can help your brain to do what it needs to do in response. So there is a specificity to the protein receptor itself on a cell. So this would be considered a target cell for this particular chemical messenger, not for this one and not for this one. So if you want a response out of this type of cell, a different type of secretory cell is going to have to put out a different chemical messenger or hormone that could fit the shape of this particular protein. Okay, so there are a lot of different signal transduction pathways then. We talked about the idea that some of the pathways will be initiated by water-soluble messengers or hormones. Some of them are going to be started by lipid-soluble messengers. So lipid-soluble messengers can diffuse through the plasma membrane. They have intracellular receptors. Instead of those receptors that sit on the outside of the cell, these receptors are inside of the cell. The signal receptor complex bind directly to recognized sequences in the DNA, and they can alter gene transcription. What is gene transcription? Remember, that's where in the process of making proteins, that's the first step in creating the mRNA that will then go out to the ribosome and create the protein. This is lipid-soluble messengers have a slower response in comparison to the receptor activation by water-soluble messengers, but it is a sustained response over time instead of a bam, you're done response. And that should make sense. So if this is a capillary and you have this lipid-soluble hormone that's going to pass through the plasma membrane, it's going to pass through the nuclear membrane, and it's going to attach to its receptor on a protein that will initiate the process of DNA transcription of a gene. So you make the mRNA travels to the cytoplasm, ribosome creates the protein, and that is considered your cellular response. Okay? Again, takes longer because we've got to diffuse through the plasma membrane, diffuse through the nuclear membrane, attach to our receptor, start the process of transcription, which takes time, start the process of protein synthesis, which takes time before we get to a cellular response. So those are lipid-soluble messengers. How do you regulate a receptor? How do you determine whether a receptor is going to respond or not respond? Downregulation is one way to regulate receptors, and that's where there is a smaller number of receptors on the target cell. This decreases the cell's responsiveness by desensitizing the cell to the messenger. Downregulation is aided by a process called internalization, entry of the messenger receptor complex by endocytosis, and degradation or storage for future use. Okay, so downregulation is going to happen on a target cell that doesn't have very many protein receptors on its plasma membrane. Okay, and that should make sense because the more receptors you have, the more hormone complex systems that can be made, and the higher the response when it comes to the cellular response. Upregulation, on the other hand, is where you have a larger number of receptors on the target cell. So that means it's going to increase the cell's responsiveness because more of those hormones are going to be able to make the complex that then is internalized and stored for later use. So you're going to have more of those internalized complexes that are ready for a cellular response at a later date. There is continuous synthesis and degradation of receptors, so altering the number of receptors is actually a common event. And that would probably, that should make sense because the receptors are made out of proteins. Proteins can be remade and recycled and reused as needed as they start to break down or are affected by negative environmental conditions like changes in pH, changes in temperature, the presence of pathogens, etc. So there has to be that ability to recycle as a response. So now let's take a look at pathways initiated by those water-soluble messengers. So the water-soluble messengers are those hormones that are going to attach to the integral protein receptors on the plasma membrane. Water-soluble messengers require second messengers to carry out their effects because they cannot penetrate the plasma membrane. There are a lot of different types of receptors. Ion channels are one example. G protein coupled receptors are another. And intrinsic kinase receptors are a third type. Different types of receptors activate that signaling cascade that happens inside the cell itself. So when we were talking about in our signaling lab, again, inside of the taste bud, different signaling cascades happen or processes happen as a result of the binding of sweet to sweet receptor, bitter to bitter receptor, sour to sour receptor, etc. Receptors can activate downstream mediators, which affect DNA transcription, but they also have a lot of other effects in the cell. So these are the ones, simply said, they are more complex systems, okay? They involve more steps than the lipid soluble messengers do. However, they produce a faster response that does not last as long as the lipid soluble messengers. So here's our first example of a type of water soluble excuse me, a type of water soluble system, and it's called a ligand gated ion channel. And we've seen the word ligand before, and that just simply means the chemical messenger that's going to attach to the receptor. Activation of the receptor by the ligand, so the hormone or whatever chemical it happens to be, results in the change of the receptor, so it forms an open channel. These types of channels are called gated, and they're gated ion channels because they are moving one or more types of ions through that channel. it often results in a change in the membrane potential. And we're going to talk a lot later about when you change the membrane potential of the cell, you are initiating an electrical response that can then be sent to the nervous system. Okay, so here's our first messenger that's going to sit in the receptor. Okay, so right now the channel is closed, but when that messenger sits down in its, and you can see when it sits down in this particularly shaped receptor spot or bonding spot, it causes a slight change that bumps open that channel. And if calcium goes through this open channel from the outside of the cell to the inside of the cell, and it's going to take more steps, okay, to be able to get to the cellular response, but that is initially how that ligand-gated channel works. So your ligand sits down in the binding site, it changes the receptor shape so that the ion channel opens, ions are allowed to pass through, which initiates other steps that are going to cause the cell to respond. And again, this is a type of water-soluble, one of the many types of water-soluble pathways. Sometimes receptors can, sometimes receptors can function as enzymes. And one of the things that's going to happen in this kind of process is phosphorylation. Now let's think about what that term would mean. We know that phosphates from our last chapter are a group that can be added or subtracted, for example, from ATP to release energy or to store energy. Phosphate, anytime you add a phosphate to a space or to a chemical, it's called phosphorylation. So enzymes can phosphorylate the receptor itself as the receptor phosphorylates some of its own tyrosine residues. And tyrosine is an amino acid. The newly created phosphotyrosines on the cytoplasmic portion of the receptor then serve as another docking site for cytoplasmic proteins. Okay, so this is where that second messenger thing comes in with some of these water-soluble processes. The docking proteins then bind and activate other proteins, which in turn activate one or more signaling pathways within the cell. All the pathways involve activation of cytoplasmic proteins that are started by adding that phosphate. So let's take a look at it. So our first messenger sits in the receptor, causes a breakup of ATP. So if you cut off one of the three phosphates, you get a phosphate that will bind to the tyrosine kinase, which is an enzyme. So this enzyme is going to speed up the process of that phosphate binding to the docking protein. So the docking protein is sitting in the cytoplasm. The ATP is floating around the cytoplasm. Why? Because cellular respiration made it in a mitochondria from your food, and it's ready to break off this PO4. This enzyme helps to bind that because enzymes speed up processes by lowering the amount of energy that is required to get it done. And then this docking protein becomes the initiating step in a multiple step process to get the cell to respond. Okay, how is this similar than the first process? You have that first messenger that sits in the receptor and changes its shape. That's what happened with the ion channel one as well. But with the ion channel one, you opened up the ion channel over here. This time, you're activating an enzyme that is going to then cause a new protein complex to start the process. Whereas in the ion channel 1, you open up that channel so the calcium that passes through can start a cascade of processes that will get you to a cellular response, all initiated by that water-soluble messenger that sits on the outside of the receptor in the plasma membrane. So that's a second example. We have receptors that interact with cytoplasmic, okay, Janus kinases. Anything that ends in an ASE, we know to be an enzyme, okay? These receptors do not have intrinsic kinase activity, but have separate cytoplasmic kinases called, and we call them JAKs, associated with the receptor. And we'll look at this picture in just a minute. So the receptor itself does not have, what we're saying here is the receptor itself does not have the enzyme embedded in it. It is instead associated with the receptor. Binding of your first water-soluble messenger to the receptor causes that shape change, and it activates the JAK enzyme. Different receptors associate with different JAKs, and the JAKs phosphorylate different target proteins, similar to the enzymes we talked about before, but many of which act as transcription factors. What's a transcription factor? Transcribing mRNA from DNA so that we may start the process of protein synthesis. So JAK activation results in the synthesis of new proteins, which mediate the cellular response. Okay? Signaling by cytokines, proteins secreted by cells of the immune system, occurs mainly receptors linked to JAKs. So what this last statement says is what's different between a general attached enzyme to the receptor that we looked at in the previous system and this system is that this is associated with immune response and the other enzyme systems are not necessarily associated with immune response. So here we go. So here's our complex here. And remember in the last picture, that tyrosine kinase, it was inside of the receptor, right? Okay, this time the kinase, the JAK, is a separate structure, a separate protein. It's still an enzyme, but it's a separate protein attached to the bottom of the receptor. Okay, so your water-soluble messenger sits, changes the shape, activates the JAK. The JAK then activates the process of phosphorylation of the protein. So we're kind of combining some things here. So we're breaking up the ATP, we're adding the phosphate to it, and then that again is initiating the multiple steps to get a cellular response. Okay, so far we've talked about the ion channel that responds to water-soluble messengers. We've talked about the enzyme kinase response to water-soluble receptors, and we're talking about the JAK, J-A-K response to our water-soluble messengers. Now we're up to the G protein coupled receptors. And I believe, if I remember right, one of the processes inside of your taste bud, one or two of them, were G protein coupled receptor reactions. You might want to go back and look at that tulip-shaped picture from that lab to double check me. To see if I'm right about that. But I believe a couple of those processes of response to taste, to a tastent, are G protein coupled. So G proteins are protein complexes that are bound to inactive receptors on the cytosolic surface of the plasma membrane. Okay, so how's that different than what we've talked about before? Cytosolic surface of the plasma membrane means on the inside surface. G proteins contain 3 subunits, an alpha, a beta, and a gamma subunit. The alpha can bind to GDP and GTP, which are uh, energy particles, similar to ATP and ADP. Okay, so they're energy packets. Think of them as energy packets. The binding of a first messenger, so your water-soluble messenger, to the receptor changes the shape, and the activated receptor increases the affinity of the alpha subunit of the G protein to attach that GTP energy particle to it. When bound to the GTP, the alpha subunit dissociates or moves from the other subunits, beta, of the trimeric G protein. Okay? The dissociation allows the alpha subunit to link up with another plasma membrane protein. So it could be the ion channels that we talked about or an enzyme. So these ion channels and enzymes mediate the next step that lead to the cell's response. They are the most numerous type of receptors and have a large variety of pathways associated with them. So let's take a look at the picture because we used a lot of words there. Okay, so can you see the difference now? So this receptor and its hormone are not going to cause directly the cell's response. What they're going to do instead in this fluid mosaic plasma membrane that we've talked about, your water-soluble messenger sits in the receptor, changes its shape. By doing that, it allows this alpha to grab on to GTP. I'm sorry, it allows this alpha part. So this is the G protein, okay? 3 subunits. Alpha has GDP attached to it. When this is changed in shape, it adds a phosphorus to the GDP, and that gives it the energy that it needs to move away from the other 2 parts of that G protein complex. It attaches to an effector protein, which can be either an ion channel or an enzyme. When it does that, it's going to change the shape of this protein, okay, and allow either an influx of ions, which is going to change the membrane potential, because that's what an ion channel does, or it's going to stimulate the enzyme that is attached to this protein, whether it is a JAK enzyme or the tyrosine kinase that we saw before. Again, that makes your secondary messengers, that phosphorylation of the proteins, and then the multiple steps towards cellular response. Okay, so this is a more complex situation, but it is also a more adaptive because of that. Because this one connection can initiate different types of responses on different types of proteins using the same messenger, it makes it more adaptable and capable of getting that cellular response just using this first messenger to initiate the process. Whereas the ion channel needed a particular messenger, the enzyme needed a particular messenger. Here, we have one type of messenger that can affect different types of effector proteins. Adenylate cyclase and cyclic AMP. So here's another second messenger system that uses, not exactly sure how I go back. that uses the G protein. So we'll just skip that last. I can talk my way through this. So we're going to begin on the left-hand side over here with something that looks similar, okay? Um, the difference in this picture and the last system that we went through is that here, adenylate cyclase, so we have a protein that has ASE, that has an enzyme embedded in it, right? Okay, we've seen that before. A protein that had an enzyme, the JAK, attached to the bottom, and then we had one that had a different enzyme that was a kinase that was attached here. Now we've got a cyclase, so just a different type of enzyme. The G protein is that process of the hormone attaching to the receptor changing shape, the phosphorylation of GDP that moves the alpha to initiate a change in the next protein. Okay, so your first messenger is here, and then that enzyme speeds up the process of turning CAMP dependent protein kinase to an active form of it by using energy from ATP. Okay, so CAMP is a secondary messenger inside the cytoplasm or cytosol that is stimulated by this enzyme to release some energy from ATP, also floating in the cytoplasm, to attach to another enzyme that will then become an active form that can phosphorylate a protein and cause a cellular response. So let's try this again from the top. The first messenger hormone changes the shape of the protein receptor, which initiates the breakoff of the alpha from the G protein, which changes the shape of the protein that has a cyclase in it that prompts this process, okay, to turn a secondary messenger into an active enzyme that will help to phosphorylate a protein to give a cellular response. So remember, I said from the beginning, from the water-soluble beginning of this discussion, that we were just going to get more complex and more complex as we went. CAMP action stops when it is broken down by AMP. And there's an enzyme that has to speed that process up, as in almost all cases. But that CAMP action of phosphorylating a protein and causing a cellular response is stopped by a molecule that will terminate the phosphorylation of any more proteins. The cellular concentration of CAMP can be changed either by altering its rate or breaking it down. Caffeine, the active ingredient in coffee, and theophylline in coffee and tea are stimulants that work partly by inhibiting the breakdown of CAMP, prolonging the actions of CAMP within cells. So if the action is to phosphorylate proteins and to cause a cellular response, then caffeine coming in and not allowing the enzyme that would break down the CAMP within the cell allows that process to be heightened and to continue on and get a cellular response that is ongoing. So all we're showing here is an ATP, and we know that because there's 1, 2, 3 phosphates attached to an adenine sugar, okay, adenosine 1, 2, 3 triphosphate. So ATP and adenylate cyclase, that enzyme that we were talking about, gives off energy and allows CAMP to attach to water and this enzyme that breaks down the CAMP into AMP, which terminates that process of the CAMP phosphorylating anything else, and being able to continue the protein cellular response. Not all G proteins stimulate CAMP formation. Some actually inhibit adenylene cyclase from working. So what we're talking about here is that some G proteins, when we're looking at that purple adenylene cyclase protein in the plasma membrane, sometimes G proteins can actually stimulate them, because these types of receptors are associated with a different G protein known as GI, which is inhibitory. Activation of GI causes the inhibition of that next purple protein. The result is decreasing the concentration of CAMP in the cell, and thereby the phosphorylation of those key proteins that are going to cause the cellular response. So this is kind of focusing on the idea of cellular responses needing to stop. There has to be something that causes a cellular response to stop. And there are a lot of different ways for that to happen. Because remember, we're trying to temporarily take something out of homeostasis, okay? The level of whatever that cellular response is in the cell, okay? Whatever that involves, the level of proteins that provide us with a cellular response has a set point where the cell is not causing a response that's going to be interpreted by the brain. So in a normal day, or in a normal time period when that cell does not have to respond to anything, it has to maintain the set point of that particular protein. So if this process were started simply by the hormone sitting down in its binding site, and it never stopped, either because that protein was broken down, or, for example, this process where you have an inhibition of the second step of the process, then the protein level that causes that cellular response would be too high all the time. And that's a positive feedback mechanism. And we don't need that, because we don't want that cell to be in response mode when it's not necessary. On the flip side of that, there are times when we need amplification of of any kind of substance. And so when we're looking at that G protein CAMP activation, okay, where a first messenger attaches to the receptor, and then that G protein, which they don't picture in here, is going to initiate the CAMP to initiate the enzyme that initiates the phosphorylation of products. Sometimes the signal needs to be amplified, and sometimes that is a good thing. If you have, for example, if you're trying to respond to something that requires you to make a very, very quick movement, you've stepped on a nail, for example, and you need to respond quickly, you want a signal amplification to get you to respond, to get your brain to understand that the muscles in your leg need to flex so that you can pull that leg away from the offending nail and be able to start the process of healing. So that's where signal amplification might be needed. So going back to the ion channels can be controlled by G proteins as well. An ion channel can be the effector protein for a G protein, and it can be directly or indirectly regulated. The ion channels can either be opened or closed, In direct regulation, the G protein interacts with the channel without any second messengers being involved. In indirect regulation, a second messenger pathway is involved. pKa phosphorylates a plasma membrane ion channel, thereby causing it to open. So these G proteins are pretty prevalent and pretty darn necessary for either inhibiting or advancing or initiating these pathways that we're talking about to get cell response to a water-soluble substance. Okay, so in this picture, it's just kind of a summary of some of these things that we're talking about, focusing this time on the CAMP method of cellular response. So, excuse me, um, CAMP protein kinase can affect other enzymes. It can affect ion channels. It can affect endoplasmic reticulum, so that transport of substances after protein synthesis is initiated will be able to be sent out in vesicles and sent to where it needs to be. It can help with making microtubules, which are a part of transport, secretion, cell shape changes. So when we're talking about cellular responses, okay, so in all the previous slides, it always just says in red down at the bottom, cellular response. What are some examples of cellular responses? Some examples of cellular responses are causing an enzyme to initiate lipid breakdown, causing an enzyme to convert glycogen into glucose, causing the DNA to transcribe a gene into mRNA and make a protein, causing the endoplasmic reticulum to package those proteins, send them out for use somewhere else. So this slide is just kind of showing how by using different methods, it can cause a lot of changes, cellular changes in a cell. In many cells, so we've talked about secondary messengers, and CAMP was one of those. CAMP was a secondary messenger that we've already talked about. In the ion channel water-soluble signal transduction pathways, the calcium ion can come in when the ion channel is open. It can also then function as a secondary messenger. It can either be increased or decreased in the cytosol to elicit a cellular response. Okay, and we said that positive ions flowing into the cell can change the membrane potential. So that's what we mean by a cellular response that has to do with a charge change within the cell. It also has direct actions on other signaling proteins. By means of an active transport system in the plasma membrane, so active transport requires energy to pull calcium in, it is maintained at an extremely low concentration in the cytosol. There's always a large electrochemical gradient favoring diffusion into the cytosol, because if you have a low amount of calcium in the cytosol, you're going to have a higher amount outside of the cell. Therefore, when things move from a high to a low concentration, that's the natural diffusion process. How do they get in there? Through those channels found in both the plasma membrane and in the endoplasmic reticulum. So mechanisms by which cellular response is related to calcium concentration is you can have receptor activation. So plasma membrane calcium channels open in response to a water-soluble hormone. Calcium is released from the endoplasmic reticulum. This can be mediated by another type of protein. Active calcium transports out of the cell. Transport out of the cell is inhibited by a second messenger. So that's one way that you can keep the level of calcium where you want it to be able to get a cellular response, or you can open those voltage-gated calcium channels. So if you're needing to increase the amount of calcium in the cytoplasm, which is usually very low, these are things that can be done to make that process happen. So it can be released from the ER, the endoplasmic reticulum. It can come in through voltage-gated channels. It can come in through calcium-specific channels. Or it can be transported out of the cell when the calcium level is too high, following some sort of a signal transduction pathway that opens those gated channels. It can be transported out of the cell. It can be inhibited from transferring out of the cell by a secondary messenger that keeps it in there once it goes through those calcium channels. Major mechanisms which can increase cytosolic calcium concentration induces the cellular response. So calcium binds to calmodulin, which changes shape. So calmodulin is the plasma membrane or the plasma membrane protein that calcium binds to, which changes shape and activates this, allowing it to activate or inhibit a large variety of enzymes and other proteins, many of which are kinases. Okay, so let's go back to the enzyme protein transduction pathway that we talked about, and there was a tyrosine kinase enzyme. So there are other different types of kinases which can be embedded in the integral protein that when calcium binds to it, it causes activation. Calcium combines with calcium binding proteins other than calmodulin and can alter their normal functions as well. A stimulus can alter the steady state by influencing the active transport systems and or the ion channels, resulting in Ca concentration. So there are calcium channels in the plasma membrane that are open directly. Those are voltage gated. Extracellular calcium entering the cell via these channels can, in certain cells, bind the calcium-sensitive channels in the endoplasmic reticulum and open them up. So in this case, we're talking about calcium as a secondary messenger. So it gets into the cell, and then it starts behaving as a secondary messenger that's going to start another process within the cell. Besides the changes to the membrane potential, calcium also binds to a lot of proteins that are already in the cytoplasm, altering their shape and activating their function. And calmodulin was the one that we talked about before. So calmodulin can lead to phosphorylation to activate or inhibit proteins involved in the cellular response. So let's take a look at the picture because that's a lot of words. So here we have our first messenger, water-soluble hormone, sitting in the receptor, either increasing calcium via the plasma channels for that. So there would be an ion channel here that would allow calcium to come in, and or a change in this receptor can cause the process of the endoplasmic reticulum releasing calcium that is already within the cell, already in the intracellular fluid. Okay, but in any case, it's going to increase the amount of calcium that is in the cytosol. This calcium can attach to calmodulin, activate it, which activates a protein kinase that then phosphorylates proteins and starts that cellular response. Okay, so the calcium acts as the secondary messenger. And the calcium can either be brought from the outside via an ion channel that opens due to this first messenger, or the change in this causes a release of calcium from the endoplasmic reticulum that's already in the center. But in any case, calcium is going to serve as the secondary messenger instead of CAMP, which was our other example. So there are other important secondary messengers. So here's your calcium that we talked about. We talked about the CAMP. We talked about CGMP. These two we didn't talk about specifically. I mentioned the IP3 protein. I'm not as concerned that it's a different mechanism, but in terms of what they do, but they are just chemicals that serve as secondary messengers within the cytoplasm. Some other messengers are called icosanoids, and they're a family of molecules produced from the polyunsaturated fatty acid arachidonic acid, which is present in plasma membrane phospholipids. So we know that the plasma membrane is made up of phospholipids. Inherently, they contain these types of messengers. And you do not need to know the specific names of these. They're generated in many kinds of cells in response to extracellular signals. The synthesis of this type of messenger begins when a stimulus binds to its receptor. So that stimulus is going to be some sort of a protein, binds to its receptor and activates PLA2, an enzyme in the plasma membrane of the stimulated cell. This is no different than what we've seen before, okay? So what's going to happen is we're going to have a water-soluble substance, which serves as the stimulus, is going to bind with PLA2, a receptor that will activate the PLA2 enzyme in the plasma membrane. The PLA2 splits off the icosanoid from the membrane in the phospholipid, and then it can be metabolized into different processes. Synthesis of these types of eicosanoids then is enzyme mediated. The enzyme being PLA2. So remember, we had the ion channels and we had enzyme mediated. So this is a different type of enzyme. It's not a JAK and it's not a kinase, okay? It's a phospholipase, so it's a lipase enzyme. They can act as intracellular messengers once they're in there, so a secondary messenger, but more often they're released immediately and they act locally. After acting, they're quickly metabolized or broken down and inactivated. They have a lot of different effects on the cells, but particularly on blood vessels and inflammation. Many of the most commonly used drugs influence their pathways. So aspirin and NSAIDs inhibit a enzyme that that causes inflammation, and by doing so, they decrease inflammation in the body and therefore decrease pain signals associated with it. So this is just a picture of how it can be made. So we were talking about the fact that your first messenger sits on the receptor, changes its shape, causing that enzyme to release the acid that will then follow different pathways here's the cellular response down here. So this particular type of acid from the membrane phospholipid can be changed into different types of icosanoids icosanoids. These are three different icosanoids, and they will have different cellular responses depending on which cell they're in. Again, cessation. The importance of cessation is to prevent chronic overstimulation of a cell by a messenger. It can be detrimental to the cell. We need to bring the cell, just like everything else, back into its homeostatic balance. Yes, we want a cellular response temporarily, but we don't, in most cases, want that cellular response to be upheld for long periods of time. So there's many ways that cessation can occur. That first messenger that sits down in its binding site can be broken down by enzymes. It can be taken up by other cells and destroyed, or it can diffuse away from the area. Then, once you have that first messenger destroyed, the concentration of the second messenger decreases, and there's a cessation of the cellular response. In addition to the removal of the first messenger, so if that's not done, the receptor can also be inactivated. It becomes chemically altered, which can decrease its affinity for that messenger, so the messenger is released and diffuses away. Phosphorylation of the receptor can prevent further G protein binding, which means that that secondary protein that the G protein binds to, that alpha protein binds to, will not bind, and therefore the secondary messenger will not be made. Plasma membrane receptors may be removed when the combination of the first messenger and the receptor enters the cell by endocytosis. So that entire receptor complex, the first messenger and the protein itself, can be grabbed and brought into the cellular environment and broken down by lysosomes. So I know this was pretty intense. Thank you for staying with me. I hope it's helped you to kind of, the bottom line for this is we have pathways that stimulate each of our cells that are started by either lipid-based messengers or water-soluble messengers. The lipid-based messengers are much simpler processes. They can be sustained for a longer period of time. The many different water-soluble pathways, such as opening of ion channels, G protein structures, CAMP structures, etc. All of those are more responsive, but they are also more complex because they need secondary messengers to help to support the process of producing a cellular response. Let me know if you have any questions.